Hello again, CAPE students. I'm Alison Peart, and this is Biology for Grade 12. Last week, we looked at biological molecules, specifically water. We'll continue in that same vein, but with a different biological molecule. Today, we delve into carbohydrates. So let's get into it. Let's get into our lesson. Today, we'll look at carbohydrates, and the specific objectives are: on completion of this section, you should be able to describe the structure of the ring form of glucose, state the difference between the alpha and beta glucose, describe the structure of sucrose, and explain the difference between the structure of glucose and sucrose. In addition, we're going to be looking at more complex carbohydrates, the all-important starch, glycogen, and cellulose. We're going to look at the whole biological principle of structure relating to function. Structure presenting certain properties which confer the ability for these molecules to perform specific functions. And so, we're going to move on now to look at carbohydrates as organic compounds that have the following properties. First of all, they contain the elements carbon, hydrogen, and oxygen. I'm sorry. Now remember, last week we spoke about the four important elements that constitute biological molecules: carbon, hydrogen, oxygen, and nitrogen. In the case of carbohydrates, however, the nitrogen is absent. And so you have the general formula of carbohydrates being CX, open bracket, H2O, close bracket, subscript Y. And it's interesting because the name carbohydrate is derived from the fact that it constitutes the carbon molecule, atom for carbo, and hydrate because hydrate represents water. If you notice that in the general formula, the percentage or the ratio of hydrogen to oxygen atoms is always 2 to 1, as in the case of the molecule of water, which is H2O. In addition, carbohydrates are categorized according to their structure. So we have the monosaccharides, and remember the prefix mono means one. The suffix saccharides means sugar. So a monosaccharide is a simple sugar, for example, glucose. There are other monosaccharides, and we discuss that later. We also have the disaccharides, and the prefix di means two. So 2 sugar units, like in the case of sucrose. And then we have the polysaccharides. Now, poly means many. So many sugar units, for example, starch, glycogen, and cellulose. So let's look in more detail, in greater detail, at the simple sugars. The simple sugars are monosaccharides, and remember I spoke about the formula: C subscript X, open bracket H2O, close bracket Y, in which X represents or is 3, 4, 5, 6, or 7 carbon atoms. The glucose with the formula C6H12O6 is one great example and a very common one of a simple sugar. Now, the glucose molecule will exist as a straight chain or straight chains and as rings, but most often glucose exists in the ring form, which is far more stable. Now, there are 2 ring forms that are dependent on the position of the H and the OH groups about the carbon atom at position 1. And I want you to remember this because it's important in terms of how the glucose molecules bond together to polymerize or form macromolecules. Now, the 2 forms are named alpha, with the OH below the ring, and I show you that in a moment, and beta with the OH above the ring. Now, these 2 forms of glucose are polymerized, or they link to form macromolecules with very different properties and roles. So let's have a closer look. If you, at first glance, they seem to look identical. In fact, they have the same molecular formula, but different structural formula. If you look carefully, you'll see that here the OH, as had been said before, is below the ring at the carbon 1 position, whereas in the beta glucose, it's above the ring. And if you notice, I have numbered the carbon atoms for you, which would be at each point. So this is carbon 1, carbon 2, carbon 3, carbon 4, carbon 5, and carbon 6 is branching off from carbon 5. If you notice that, in the case of beta glucose, the OH group is above the ring. So you have the carbon 1, 2, 3, 4, 5, 6, and the OH group is above the ring. And please remember the structural makeup of those two forms of glucose. We call them isomers. Now, glucose has 6 carbon atoms, and it's an example of what we call a hexose sugar. Now, remember, hex, that prefix means 6. In the same way that tri means 3, yes? Penta means 5. Hex means 6. And of course, hepta, like heptathlon in the Olympic Games, heptathlon, 7 events. Heptose would be a 7 carbon sugar. Now, there are other hexose sugars that like fructose, which is fruit sugar, and galactose, which is actually the monosaccharide that combines with glucose to form the disaccharide lactose. Now, in days gone by, there used to be baby food that we call lactogen. I don't know if you students would know about that, but if you ask your parents, they would tell you about the popular lactogen. Lacto means milk. So lactose, milk sugar, and galactose is the monosaccharide that combines with a common glucose molecule to form the disaccharide lactose. Now, glucose is a polar molecule, and remember last week we spoke about polarity and that water is a polar substance, and polar substances like sodium chloride is very soluble in water. Glucose, we know, is very soluble, it's very water soluble. As a matter of fact, in days gone by, you used to have little babies, newborn babies, being fed with glucose because it was thought to be a very quick and efficient source of energy. It gets straight to the bloodstream in no time. And that is why it is a form of carbohydrate that is transported in the blood of animals. And in plants, it is readily taken up by cells and metabolized. Because remember now, glucose is one of the first form substances in the process, the food-making process by plants called photosynthesis. It provides a source of energy, therefore, from the process of respiration. I am not sure if you remember we used to have athletic meets, and then you might see the athletes being given doses of glucose, and they're licking the glucose, expecting to do well because it will give them a burst of energy. But alas, if you can't run like Usain Bolt, you might not very well make it to the line. So glucose is a very quick source of energy from the process of respiration. It is able to be polymerized to form polysaccharides for energy storage. And we'll speak about that when we deal with the complex carbohydrates later. It's also used as a raw material to make compounds like disaccharides. So glucose and fruit sugar fructose will link together or bond to form sucrose. And sucrose is like cane sugar or the brown sugar that we use in our homes to sweeten things. Other common monosaccharides that you'll come across are the pentoses, and those are important in the formation of the all-important DNA molecule. And those would have 5 carbon atoms, whereas the trioses would have 3. Now let's have a look at the monosaccharides, how they join together to form complex sugars known as disaccharides. The diagram below, and I'm going to show you in the next slide, shows how this happens in plant cells that make sucrose from glucose and fructose. Now, in the reaction, which is called a condensation reaction, a molecule of water is removed so that you form an oxygen bridge, and I'll show you that in a moment, between carbon 1 of the glucose molecule and carbon 2 of the fructose molecule. That the bond that is formed is known as a glycosidic bond, so named because a glycoside is a ring-formed sugar compound. And the type of reaction, as I said before, is a condensation reaction because water is formed. You know, in the mornings when you get up, all the plants have droplets of water on it. That's due to condensation overnight from water vapor to liquid water. So we call it a condensation reaction because the molecule of water is formed. Now the glycosidic bond, please note, it's a type of covalent bond. And remember last week we learned that the covalent bond is very strong as compared, for example, to the hydrogen bond. Now I want you to take a closer look at the diagram there. It shows you the molecule of glucose being added to the molecule of fructose and the resultant sucrose molecule plus water. And remember I mentioned the oxygen bridge, which is formed right there. That's the oxygen bridge, and the water molecule is given off to confer on it the term condensation reaction. In fact, sucrose is never formed in that simple way. The reaction is far more complex, but for our purposes, we're going to look at it in this simple form, and the formation involves, remember, the elimination of water as shown. Sucrose is formed by plants and it's transported in the phloem tubes through the plant from what we call the source, where it is produced by the process of photosynthesis, to where we call the sink. Those are the areas where you'd probably have them being used in metabolic or chemical reactions, or maybe stored in the fruits, the seeds, the flowers, the roots, the stems. All those are the storage areas. Now, sucrose is also a polar and therefore water-soluble substance, but it's not as reactive as glucose and fructose because the aldehyde, and you would have learned about the aldehyde and ketone groups from your organic chemistry classes in CSEC biology and chemistry. So you would know about those compounds, and those aldehyde and ketone groups form the glycosidic bond, and they are not available to react. So the lack of reactivity of the sucrose molecule is important in its role as the transport form or in translocation of produced substances in the source, that's the leaf of the plant, to where it's going to be stored elsewhere in the plant. So there's an advantage there to have a less reactive sugar for transport in plants, as the transport is slower than that of glucose in animals. I just want to mention the fact that if you notice, here we have the OH in red and the H in red. That is going to be linking to form the water molecule, and then this oxygen will form the glycosidic bond, which is really at the oxygen bridge. Just to point out to you, and if you notice, the carbon atoms are numbered as well for your better understanding. We're going to move on now to the complex carbohydrates. Glucose and other monosaccharides, they are referred to as monomers. Now remember, the prefix mono means one. So one unit will combine with another, with another, with another, until you have polymers or macromolecules with many units known as polysaccharides, and these are used for energy storage and for making cellulose for cell walls, and that would be the other polysaccharide that we're going to be looking at. And just to remind you that when we have the digestive process in man, for example, and we take in starch, cooked starch, it is going to be digested, absorbed, and taken via the hepatic portal vein to the liver. Now, in the liver, you're going to have, if you have excess glucose molecules produced from a meal, maybe you ate a lot, or you ate a lot of sugary substances, and you are in a resting phase, so you're not using up the energy, you'll find that that excess glucose would have to be stored, and it's stored in the form of that biological molecule called glycogen. We're going to discuss that a little later. But polysaccharides, therefore, are made from many monomers, and the previous figure shows a glucose monomer added to the end of an existing chain with the formation of a glycosidic bond. And if you notice, I keep repeating the information because repetition is the mother of learning. And I want you to really understand. There are some of you who would be way ahead and are quite all right with what is being taught. But there are some students who are not as ready, and they're the ones I have to cater to as well. So don't you mind the repetition, because it is the source of learning. Now, the glycosidic bonds that form between carbon 1 and the end of the growing chain, carbon 4 of the glucose monomer that is being added, they are known as a 1,4 glycosidic bond. So, for example, if you consider this as a polymer, and each of these beads would be a glucose molecule, and I'm taking the first one. So carbon 1 would be at this side, then carbon 4 would be of the next monomer that I'm going to be adding would be closer to it, and so it's a carbon 1 connecting with carbon 4 to form the glycosidic bond. And that is why it is called 1,4 glycosidic bond, because of the carbon atoms to which the bonding or the link is going to be made. If all the glucose monomers are added in this way and you get an unbranched chain, the unbranched chain is formed, then the branching point is formed by adding a glucose monomer to the carbon 6 on a growing chain. And let me just go back a bit to show you where, carbon 6 would be here, and so the link would be made here if you want to have a branching aspect to the chain. And we're going to discuss the complex carbohydrates which have branching chains. So the previous figure showed that the glucose monomer added to the end of an existing chain with the formation of a glycosidic bond. Now, the type of glycosidic bond that forms to make these branching points is not a 1,4 glycosidic bond, but a 1,6 glycosidic bond. And it's simply telling you which carbon atoms are connecting to form that glycosidic bond. From the first monomer added, another chain can form with more 1,4 glycosidic bonds, joining the glucose monomers together. Now, there are 3 energy storage polysaccharides: amylose, amylopectin, and glycogen. Now, you may remember when you did your CSEC biology that when we ingest cooked starch, it is digested by an enzyme called salivary amylase. Now, if it's starch and you usually have the enzymes being named for the sugar that it's acting on or the substrate that it's acting on, then if it's sucrose, acted on by sucrase, lactose by lactase, then if it's starch, then it should be starchase. Not really. It's actually salivary or pancreatic amylase. Why amylase? Because the starch molecule, it consists of amylose and amylopectin. They are forms of starch, and I want you to remember that. So because amylose and amylopectin are forms of starch, then the enzyme that acts on it is called amylase. Now, glycogen, the third one, is also an energy storage polysaccharide, and I had discussed that earlier. Amylose and amylopectin, as I mentioned, are forms of starch. Glycogen, which is sometimes referred to as animal starch because it is only found in animals, then the glycogen is similar to amylopectin, but it has more 1,6 glycosidic bonds than amylopectin, and therefore has many more branches. Now, glycogen, as I mentioned, called animal starch, and the 3 energy storage polysaccharides are made from alpha and beta glucose monomers. I think that's incorrect. It's alpha glucose monomers alone, because cellulose, which is the third polysaccharide complex carbohydrate that we're going to study is a long chain molecule made from beta glucose monomers only. It is not used for energy storage, but for making the cell walls of plants. And if you remember, we always speak of the cellulose cell walls of plants. If you notice, each of these, it's like the B that I have there, each of these is a glucose unit that's linked together by glycosidic bonds. And these are the beta glucose molecules. And then they are lined up in parallel forms. And if you notice here, it's almost like, I'm not sure if you know what a crocus bag is like. You know, nowadays you children aren't so familiar with these old time things. But notice that this here represents the cell wall of a plant cell. And it shows how the cellulose is structured in a linear manner. And the fact is that cellulose is a polysaccharide that performs a structural function, not for storage, as in the case of starch. Now, if you notice, starch and glycogen are branched. And in the case of starch, it is not as branching and compact as glycogen. Notice if you did the test for starch on a leaf, for example, all these dark areas here are the blue-black coloration that you'll get when you use iodine in potassium iodide solution to test for starch. And then the third image shows you glycogen, which is usually stored in the liver and the muscles of animals. Remember, glycogen is referred to as animal starch because it's only found there. Now, what I've done is to create for you a table that will encapsulate and pretty much give you a chance to analyze and to put together all the information that I've shared with you this morning. If you have a look at the headings, polysaccharides, monomers from which they are made, the type of glycosidic bonds, the structure as a result of that, the bonds and the monomers used, and then the functions that they perform. So the first one, starch, made up of amylose and amylopectin. If you notice, amylose has monomers of alpha glucose combined to form combined and linked by glycosidic bonds. The structure as a result are unbranched chain right-handed helix structure, and it's suited for energy storage in plants. Then we have amylopectin, the monomers, alpha glucose, and the glycosidic bonds involved, 1,4 and 1,6 glycosidic bonds, and you need to remember this information. The structure as a result, branched chain, and it's not a helix. You remember we spoke of a double helix, a kind of spiral that you'd see in the DNA molecule. That's what a helix is. And the function, energy storage in plants. Then we have glycogen. Glycogen is also referred to as animal starch, and the monomer involved is the alpha glucose. The glycosidic bonds, 1,4 and 1,6. The structure resulting, branched chain, more branched than the amylopectin. And remember, we looked at it. It was very dense and highly branched. And the energy storage in animals, fungi, and some bacteria. The final polysaccharide is cellulose. Cellulose constitutes monomers, the beta glucose. And the glycosidic bond would be the 1,4 glycosidic bond. So if you notice, amylose and cellulose are joined by glycosidic bonds that are 1,4 glycosidic bonds. The amylopectin and glycogen, which are quite similar, both have 1,4 and 1,6 to give branching structures. And you notice that amylose is unbranched, it's an unbranched chain, and cellulose is also an unbranched but straight chain. The cellulose is grouped together in bundles within the plant cell walls, and it has a high tensile strength, almost equivalent to that of steel, which you know is quite strong. Now, let's look again at the structure and function, the relationship between structure and function of these polysaccharides. The storage polysaccharides are insoluble in water, they are unreactive and compact. And if you wish, you can get some starch and you can add it to water, tap water and stir it, and you'll notice that there will be, it's quite insoluble and it will not form a solution as glucose would. Amylose and amylopectin, they are stored in plant cells, and starch grains and glycogen have branches. They have many places where glucose can be added or removed as required by a cell. Now, alternate beta glucose molecules are arranged at 180 degrees to each other as they are added to a growing cellulose molecule. And this gives a straight chain. Remember I said it's not a helix, which is spiral, with many projecting OH groups on both sides of the chain to form hydrogen bonds with adjacent cellulose bonds and adjacent cellulose molecules. A bundle of cellulose molecules, as I had shown in the picture, bonded together by hydrogen bonds, forms a microfibril, and that is very strong. Now, the microfibrils are arranged in plant cell walls in a crisscross pattern to provide even more strength. And if I go back to that picture, if you notice the crisscross pattern in that first lower left-hand corner where the polysaccharides in cells, you'll see the crisscross pattern of the cellulose chains. Now, to look closer at the cellulose, the structure of cellulose, that in the far left-hand corner, you'll see the plant matter. And if you should pull out the cellulose fiber, you get a macrofibril. And here we have, pulling out again, a microfibril. And when you look at the fine structure of it, these are really chains of cellulose molecules. Now, if it has this kind of structure, and this one is an example of cellulose fibers from a ponderosa pine, I have a feeling it might have something to do with the cowboys in the days of Bonanza. So cellulose fibers from a ponderosa pine might be found in the desert. The macrofibrils compose each fiber, and each macrofibril is composed of bundles of microfibrils. The microfibrils are in turn composed of bundles of cellulose chains, and the cellulose fibers can be very strong. And this is one reason why wood, for example, is such a good building material. Now, before I get to quiz time, perhaps we could recap a bit what we have looked at today. First of all, looking at carbohydrates made of carbon, hydrogen, and oxygen in various proportions. We looked at the simple sugars like glucose, fructose, and the disaccharides. Those who did the monosaccharides, we looked at the disaccharides like sucrose and the structure of it. We looked at the way the glucose molecules would bond to form more complex molecules, the disaccharides. You have also a group called the oligosaccharides, which would be those with the few monomers constituting them. Then you have the polysaccharides, which are far more complex. Poly meaning many. Polysaccharides like the starch, cellulose, and of course, glycogen. We also looked at the way the complex carbohydrates combine using the glycosidic bonds, 1,4 glycosidic bonds and the 1,6 glycosidic bonds, conferring certain special functions, certain properties that will make them suited. In the case of glucose and in the case of starch and glycogen for energy storage, whereas for cellulose it's really for the strengthening properties that would be required in plant cells. All right, so let's have a look at the questions I have for you. It's quiz time. Which of the following combinations of statements is true of polysaccharides in living organisms? So the headings are to provide energy, to form storage compounds, or to form supporting structures. And we have here 5 options, A, B, C, D, and E. A says no in terms of providing energy. Is that correct? I don't think so. We have starch providing energy. Do they form storage compounds? Sure they do. Glycogen and starch, they form storage compounds. And forming supporting structures? Of course, yes, that's correct. Cellulose. But the other 2 are incorrect, and so A would be wrong. So let's look at B. It says no to providing energy, no to forming storage compounds, and no to forming supporting structures, and that is incorrect. If you look through C, yes, providing energy, but it also forms storage compounds. There's a no there, so that could not be correct. And forming supporting structures, no as well. Therefore, C would be incorrect. D says yes to providing energy, yes to forming storage compounds, but no to forming supporting structures. Cellulose does that, and therefore D is incorrect, and the correct answer would be E, because they perform all of those functions, providing energy, forming storage compounds, and forming supporting structures. So you see, as you go through, you have to assess each of the options and make sure that you don't guess, and you use the information that you have at your fingertips to answer the questions correctly. Number 2, polysaccharides are synthesized in plants by the process of A, condensation, B, glycolysis, C, hydrolysis, D, oxidation, and E, reduction. Now, remember we said that polysaccharides are formed from their constituent monosaccharides, and when this happens, water is given off. It's referred to as a condensation reaction. Therefore, your answer is going to be A. Number 3, most polysaccharides are composed of chains of condensed, is it cellulose units, hexose units, C, pentose units, D, sucrose units, or E, triose units. So, I'm sorry. And so the answer has to be B, hexose units, because glucose, which is the predominant monosaccharide, is a hexose molecule. So B, hexose units. Benedict's test was performed on 20 cm cube sample of 4 fruit syrups. A second 20 cm cube sample of each was then hydrolyzed and tested with Benedict's solution. The table shows the masses of precipitate formed. Now, this is going to take a little doing. You have to evaluate the information given. It is asking you to state which syrup contains the greatest mass of non-reducing sugar. Now, the mass of precipitate before hydrolysis per milligram in the first column, and the total mass of precipitate after hydrolysis per milligram. A says 25 and 55. B says 50 and 70. C, 55, 55. And D, 75, 85. Now, bear in mind that in the test for reducing and non-reducing sugars, we use the Benedict's solution. And the fact is that you have to hydrolyze the sucrose, or non-reducing sugar, in this case it would be sucrose, would hydrolyze it using dilute hydrochloric acid. So the mass of precipitate before hydrolysis would be far less than that after hydrolysis. And in this case, B, C, and D seem to have a large mass of precipitate, and therefore, before hydrolysis, and therefore those could not be the correct answer. The correct answer, therefore, is A, where the difference is about 30, significantly more mass of precipitate after hydrolysis than before. So this one requires a little more a higher order operation, but don't be daunted. I know sometimes when you see figures, you feel that you're going to skip that one. Don't you do that. Sometimes they are the simplest questions on the paper. I'm going to leave number 5 for you to take a snapshot of if possible. And I'm going to ask you to do that for your homework. That one takes a little thought and a little getting used to. It says 5 disaccharides were each hydrolyzed with dilute acid. Of course, it would be hydrochloric acid. And the purified products were separated by one-dimensional chromatography. Now, remember, chromatography you would have done at CSEC level, where you would separate the dyes in ink using filter paper. I'm not sure if you remember all of that, but that's important. And so you have the spots moving at the ink moving at different speeds through the filter paper, and thus causing the dyes that constitute the ink to separate out. A similar fashion here, we have the chromatogram shown in the diagram below. And this is for sucrose, is number 1. The questions are, if spot 1 represents the products obtained from the hydrolysis of sucrose, which of the following indicates the results obtained from hydrolysis of lactose and maltose? So you're going to, those are the options there, lactose, maltose, and you're going to look at this diagram and try to analyze it, the positions of the spots, and to see which ones would be lactose and maltose, and then you make your selection there. That is for homework. I hope you have captured it. I'm going to allow you a little time to just do so, and we could probably show it afterwards. Now, the diagram shows a carbohydrate. What is the name of the bond which links the 2 units? And we have done that all morning. That's the link here. That's the bond. What is the name of the bond which links the 2 units? A, glycosidic bond. B, hydrogenic bond. C, oxygen bond. D, peptide bond. E, saccharide bond. And of course, we know it's the glycosidic bond. And this one, I'm going to ask you to take a snapshot of that. You're going to be doing that for your homework as well. Notice I'm giving you ones that require a little thought to do at home. Moving right along to the last one, I think this is, which conclusion is consistent with the results? And that you're going to take. Those are the questions for the previous slide. Of course, success is a must. Grade 1 straight A profile. That's all the time we have for today. Until then, my children, stay safe.